Welcome to the deep dive. Today we're looking at something, well, pretty fundamental. A tiny genetic switch,
Mm-hmm.
and it might just hold a key to understanding what makes the human brain,
Real.
human, and maybe even shed light on some neurological conditions.
Absolutely. We're focusing on a specific bit of DNA. It's called HAR123. And yeah, the research suggests it's a major player in how our brains got built.
Right, and this is all coming from some pretty recent work out of UC San Diego published in Science Advances.
That's the one. Groundbreaking stuff.
So our mission really is to unpack this. What does this little piece of DNA tell us about our evolution, our
Yeah.
thinking? And yeah, those links to neurodevelopmental disorders too.
Let's dive in.
So, okay, first things first. Let's set the stage. We're talking about HARs, human accelerated regions. What are they exactly?
Right. So HARs, they're specific sections in our genome. Think of our complete DNA map.
Got it.
These sections show way more changes, more mutations in humans compared to chimps ever since we split off evolutionarily, about 5 million years ago.
So evolution's been tinkering faster in these specific spots in humans.
Precisely. They're like evolutionary hotspots for us, and that's why scientists are so interested. The idea, the hypothesis, is that these HARs are involved in giving us our uniquely human traits.
Makes sense.
And importantly, some have also been linked to conditions like autism, so there's a potential connection to brain development and function, both typical and atypical.
Okay, so are these HARs genes? Like instructions for building proteins?
Good question. Mostly no. That's what's kind of fascinating. About, 96% of HRs are in the non-coding regions, the parts that don't directly code for proteins.
The so-called junk DNA, although we know now it's not really junk.
Exactly, not junk at all. Many of these HRs, even though they're non-coding, act as transcriptional enhancers.
Enhancers, like genetic switches.
Yeah, or maybe like dimmer switches or volume controls. They don't build the protein, but they tell the actual genes when to turn on, how much to turn on, and where during development, for instance. They regulate the activity.
Okay, so HAR123 is one of these enhancers. Where do we find this specific one, and how old is it?
So HAR123 specifically is this 442-letter sequence, nucleotides located inside another gene called SMG6, but in a non-coding part, an intron.
And its age?
It's ancient, really ancient. It's found in basically all mammals and even marsupials. That suggests it first appeared maybe 170 million years ago.
Wow. Guy?
But here's the twist. While it's super conserved, like almost identical across most mammals, in the human lineage, since we split from chimps, it's changed rapidly. There are 9 specific nucleotide differences between the human version and the chimp version.
9 changes in something that old and usually stable. That seems significant.
It really is. It points to strong evolutionary pressure specifically in humans acting on this enhancer.
So if HAR123 is this volume control, what's the proof it's actually, doing anything, especially in the brain?
Right. Good question. Well, first, scientists looked for chemical tags on the DNA itself, these things called chromatin marks. And the marks around HAR123 were consistent with it being an active enhancer, like signposts saying, this bit is working.
Okay, so the signs are there.
Then they did experiments in vitro, in lab dishes, using human embryonic stem cells. And they confirmed that both the human version, HAR123, and the chimp version, say R123, do act as enhancers. They both turn up gene activity.
So both versions work as enhancers in a dish. But what about in a developing organism, like in an embryo? Does it matter where they
Oh,
work?
yeah, this is where it gets really interesting. They tested this using mouse embryos. They put the human HR123 or the chimp HR123 into these embryos and watched where it activated genes.
Yeah.
The human version, HR123, mostly drove activity in the forebrain and the midbrain, regions critical for complex thought planning.
Higher cognitive functions, yeah.
Exactly. Very little activity in the hindbrain.
And the chimp version, HAR123.
The chimp version, well, it also showed some midbrain activity, so there's overlap,
Yeah.
but it was frequently active in the hindbrain and much less often in the forebrain.
So same switch, different locations. Human version favors the front, chimp version the back, relatively speaking.
That's a great way to put it. This difference in where it's active strongly suggests they're playing different roles in shaping the brain architecture in humans versus chimps.
Okay, so HAR123 influences where genes get turned up in the developing brain. How does that connect to the actual cells being made? You mentioned neural progenitor cells.
Yes, exactly. HAR123 seems crucial for making these neural progenitor cells, NPCs. Think of them as the stem cells of the brain.
The ones that become neurons or glia.
Right, neurons for signaling, glia for support. NPCs are the source. Now, when they knocked out HR123 in human embryonic stem cells,
Took it away completely.
yep, they found it really messed up the ability to make these NPCs. The cells either just stayed as basic stem cells or got stuck partway through the process.
So without HR123, you don't properly get the building blocks for the brain going.
Pretty much. It seems essential for that initial push towards becoming neural tissue and then generating those progenitors.
Is the human version better at this than the chimp or mouse versions? You said they all worked as enhancers.
But there's a difference in efficiency and maybe specificity. So the chimp and mouse versions can push human cells towards making NPCs, which tells us there's a core function that's conserved, it's ancient, but they're less efficient than the human HAR123. With the chimp or mouse versions, more cells got stuck in those earlier undifferentiated stages.
So the human one is like better tuned for the job in human cells.
It seems that way. And more than that, the human HER123 seems to regulate a specific set of genes involved in, say, cell growth and maintaining these neural stem cells, genes that the chimp and mouse versions don't regulate in the same way in these human cells.
Suggesting a uniquely human fine-tuning of the process.
Exactly, a human-specific layer of regulation.
And this affects the balance of neurons and glia too, right? The neuron-glia ratio. Why is that important?
Hugely important. The balance between neurons and glial cells is critical for how the brain processes information, for its overall health. Imbalances are linked to various neurological issues.
And what did removing HER123 do to that ratio?
They found that losing HR123 caused a significant decrease in the neuron-to-glia ratio. It shifted the balance away from making neurons.
So HER123 normally promotes neuron production relative to glia.
That's what the data suggests. And they saw this not just in the dish, but also in vivo, in the knockout mice, specifically in parts of the hippocampus, the CA3 and dentate gyrus.
Which are key for memory and learning.
Right. And they saw this effect both early in development and later, suggesting it's a sustained influence.
Okay, so we have HR123 influencing location, progenitor cells, and neuronglia ratios. Did the researchers figure out how it's doing this? Like, what gene is it directly controlling to make this happen?
They did find a direct target, a gene called HIC1. It stands for hypermethylated in cancer 1, but its role here is in brain development.
HIC1. And what does HIC1 do?
HIC1 makes a protein that acts as a transcriptional repressor, so it turns other genes off.
Okay, so the enhancer HR123 turns on the repressor HIC1. That seems a bit counterintuitive.
It does at first glance. But here's the proposed mechanism, and it's actually quite neat. HR123 activates HIC1.
Right.
Then HIC1, the repressor, goes and turns off genes that are responsible for keeping the early neural tissue, the neuroectoderm, in an undifferentiated sort of basic state.
Ah, I see. So by repressing the stay-put signals, HIC1 effectively pushes the cells forward to become neural progenitors.
Exactly. HR123 turns on HIC1, HIC1 turns off the brakes, and the cells move on to become NPCs. When HR123 is missing, HIC1 levels are lower, the brakes stay on, and NPC formation is suppressed.
That's a really elegant circuit. Is this HR123 HIC1 connection itself something special in humans?
It seems like it might be, at least in human cells. The study found that only the human version of HR123 could effectively drive the expression of human HIC1 in these early neural cells. The chimp and mouse versions didn't do it nearly as well.
Wow. Okay, so that points strongly towards a human-specific pathway for generating these crucial brain precursor cells.
It really does. A molecular circuit potentially unique to our lineage.
All right, this is all fascinating at the cellular and molecular level. But does it translate to, well, behavior, cognition? What happened with those knockout mice when you looked at how they act?
This is maybe one of the most striking parts of the study. So these HAR123 knockout mice, they looked pretty normal on the surface.
Viable. Fertile.
Yep, viable, fertile. Basic movement was fine. They could recognize objects they'd seen before, standard learning tasks. They were okay.
But there's always a but.
There's always a but. The key came with reversal learning tasks.
Reversal learning. That's about adapting when things change, right? Cognitive flexibility.
Precisely. So things like the Barnes maze or a water maze where the mouse first learns to find an escape hole or a platform. The knockout mice learned the initial task just fine. But then the researchers moved the escape hole or the platform. The rules changed. And the HAR123KO mice really struggled with this. They had trouble unlearning the old location and finding the new one.
So a specific deficit in adapting to new information. Cognitive inflexibility.
Exactly. A deficiency in cognitive flexibility, which is considered a pretty sophisticated cognitive ability.
And did they link this back to gene changes in the brain? in areas involved in flexibility, like the frontal cortex?
They did. They looked at RNA levels in the frontal cortex of these KO mice. And sure enough, they found changes in the expression of genes known to be involved in cognitive flexibility. Genes like TTR, NR4A2, COL24A1, DRD2.
Providing a potential molecular basis for the behavioral deficit.
Right. And remember the hippocampus, where HAR1 affected the neuronal ratio?
Yeah.
The hippocampus is also critically involved in cognitive flexibility. So the changes in cell ratios there could also be contributing to this behavioral outcome. It connects the cellular changes to the cognitive ones.
It's all starting to tie together. So stepping back now, what are the big implications here for understanding human evolution and maybe for neurodevelopmental disorders?
Well, for evolution, HR123 is a fantastic example. It shows how these specific accelerated changes in regulatory DNA, these HRs, could have actually built some of the unique features of the human brain after we split from chimps. It's not just theory. Here's a potential mechanism.
A concrete example of evolution acting on a regulator to change brain development.
Exactly. And for neurodevelopmental disorders, this is also really important. We already knew HRs in general were associated with things like autism and schizophrenia.
Right.
But HR123 and its target, HIC1, they sit in a specific spot on chromosome 17, a region that's known to be unstable. Deletions there cause rare but severe disorders, like Miller-Dieker syndrome, which involves major brain malformations, lissencephaly, or smooth brain.
Wow. And is there a direct link in patients?
There is. When they looked at patients with deletions in this exact region, deletions covering both HR123 and HIC1, those deletions perfectly matched up with having specific abnormalities in the brain's white matter.
So losing HR123 and HIC1 correlates with structural brain problems in humans.
It strongly suggests HR123 could be a key player in these conditions, especially since we know HR123 affects that neuronglia balance, and imbalances in those cells are linked to these kinds of disorders. It makes HR123 a really strong candidate gene to investigate further.
So this deep dive really went from a tiny DNA sequence all the way to potentially explaining aspects of human brain evolution, cognitive flexibility, and even serious developmental disorders.
Yeah.
It's more than just finding HRs, it's showing what they do.
Absolutely. It nails down the functional impact. HR123, this ancient enhancer that got supercharged in humans, it really does seem to act like a conductor, orchestrating key parts of brain development, shaping the cell types, the ratios, things that likely underpin our unique cognitive landscape.
Amazing stuff. So here's something for you, our listeners, to think about. If one small change, relatively speaking, in a non-coding bit of DNA like HR123 can have such potentially massive consequences shaping our human minds, our flexibility, maybe even our vulnerability to certain conditions.
Yeah.
What else is hiding in there?
Right, in all that vast non-coding territory of our genome.
What other fundamental truths about who we are, how we evolved, how our brains work, are just sitting there waiting for the next deep dive to uncover them? Something to mull over.